Okay, so we're going to continue our discussion about the organic compound lipids. That is one of the four that make up much of our body. And we're going to now discuss phospholipids. So these are similar to triglycerides, but instead of having 3 fatty acid chains attached to a molecule, they only have 2. And it's not connected to a glycerol molecule. It's connected to another smaller molecule that contains a phosphate group. So you can see that written here, phosphate group, and it's this little group here. So we call this region of the phospholipid the head, and it's also drawn for you kind of here in cartoon form. And we call these fatty acids, this whole region, the tail, and they're drawn for you there. Because of the properties of the phosphate region or the head region of the phospholipid, it makes the head region like to be around water. It's a water-soluble piece of this molecule. The tails, however, are made of fatty acids, and so the tails are not water-soluble. And so they don't want to be around water at all in our bodies. So what's interesting is these properties of phospholipids make them perfect for forming a double membrane that we actually have as the boundary of all of our cells. So if you think of a water balloon, right? The balloon, the rubber part of the balloon is actually what I'm talking about. That is similar to what a cell membrane would look like. And then the water inside the balloon would be all the stuff inside the cell itself. So these phospholipids, if you have a whole bunch of these, say you've got, 500 little phospholipid molecules, and you throw them into water, they will automatically assemble themselves in a way that allows the tails to be away from the water and the heads to be like touching the water and happy. So let's see what that looks like. So here you have 4 phospholipids, and here's another 4 phospholipids. And this is what they do when they're forming our cell membrane. The outer environment of our cells is very watery. The inner environment of our cells is also watery. So the heads want to be in that environment, but the tails don't because they're lipids. So the tails will face each other to try to get away from the water, and it forms this kind of lipid layer, right? And once we talk about this a little later, you'll see how this lipid layer actually controls what can enter and leave the cell, which is kind of cool. Water-soluble things cannot sometimes get through there. You have to be fat to get through. So anyway, we'll get to that another time. So we call the head portions of these phospholipids hydrophilic, which means water-loving. And the tails we call hydrophobic, which means water-fearing. All right. So now on to another type of lipid. They're called steroids. There's a number of different types of steroids. All steroids have the same core structure to their to the molecule, and you're seeing that highlighted in color here. So they call this the steroid nucleus, just because it's the core of the molecule. But what makes each one of these steroids different are these side chains coming off of the core of the molecule, right? So these side chains give this particular steroid characteristics, and we call it cholesterol. This steroid has different side chains, and it therefore has different functions. And this particular one is called cortisol. Down here you have the quote-unquote sex hormones, right? Estrogen and testosterone. So you can see they're very close to one another, but boy, do they have some different effects, huh? Yeah. All right, so and there's many other molecules that have steroids associated or are steroids, but this gives you an example of that type of lipid. And finally, prostaglandins. So I don't ask you to know a lot about prostaglandins because even though it's a single class, prostaglandins have so many various, strangely different functions throughout the body. And these are actually referred to as tissue hormones. They're produced by cell membranes in tissues throughout the body. And depending on where they are and the particular type of prostaglandin it will have a very different effect. So these are some of the functions of different types of prostaglandins. They can regulate hormones, blood pressure, digestion, immune system functioning. They can help to aid in clotting. They play a role in respiration and also in regulating body temperature. So you can see that these functions are like very different from one another. All right, so moving on to proteins. So proteins, again, are the most abundant organic molecule in our body. They form the structures in our body. They're involved in a number of different chemical and cellular reactions. There are special proteins that exist in our immune system called antibodies that help us fight against infection. Proteins build our bones and build our muscles, and oh gosh, I mean, proteins are just everywhere. So something to remember about proteins is that every protein starts off as a chain of amino acids that have been bound together. So hundreds of amino acids. So that's the very basic form of a protein, and then the amino acid chain will fold and do this and do that, and we'll talk about that in a bit. But the very basic level is amino acids, right? Once a protein's amino acid chain is folded into its mature form, its functional form, we know that the shape of a particular protein determines its function. It's kind of like a key, right? In order to open a lock, you have to have a key that has the correct shape. And it's the same with proteins. In order for a protein to do the function that's needed to be done, a particular protein has to have a very specific shape, because proteins work by binding to other molecules, and the fit has to be correct. So if that shape changes, the protein's function goes away. So don't forget that. That is super important to remember, right? Shape determines function. All right, so here's a list of all those 20 different amino acids I talked about earlier, and combinations of all of these, oh shoot, come on, behave. Oh, behave. Here we go. Again, what form those chains of amino acids and form that first level of protein structure. So, and as you remember, I hope, all amino acids have the same basic backbone. This is identical to this, and this, and this, and this, and this. These are 6 different amino acids, but what makes them different is the functional group that you see here coming off of that, that basic core part of the molecule. So, amino acids consist of a central carbon, an amine group, which is the one that has the nitrogen here, the N in it, another group over here called the carboxyl group, which has the COOH in it. So that part remains consistent for all amino acids. And they all also have a hydrogen coming off the central carbon here, and this R group, right? And so remember, the R group is that functional group. Just like with carbohydrates and proteins, and lipids, guess how proteins are formed? and broken down. Make you think about it for a minute. Hopefully you remembered that those other molecules are formed from dehydration synthesis, and they're degraded or broken down by hydrolysis. It's the same thing that happens with proteins. So here you can see this is an amino acid here, and here's another amino acid. So, oops, sorry, up here, I'm sorry. This is an amino acid, and here is an amino acid. The hydrogen is going to bind with the OH group on this other amino acid, forming water. Water will go away, and then the remainder of this molecule and this molecule will bind together here, forming what is called a peptide bond, because it's between two amino acids that are going to make a protein, and proteins are called peptides. So this is now a 2 amino acid chain that's going to begin the first part of protein structure. So anything that gets formed by dehydration synthesis can be degraded by hydrolysis. So again, you can have water come in, break this peptide bond. The hydrogen will get attached to one amino acid, the remaining OH to the other, and it separates them into 2 separate amino acids now. There are 4 levels of protein structure, and I'm going to go through this quickly because I've only got a couple minutes left of this video. So the primary structure we've already talked about. It is a chain formed from amino acids binding together, forming peptide bonds in between them. So all of these are different amino acids forming the chain. Then that chain will start to fold into one of 2 shapes. It will fold into either this kind of a funky pleated sort of shape, and we call that a pleated sheet, or it will form this coiled structure that is a helical structure, and we call that a helical shape. So the secondary level of protein structure is either going to be a beta pleated sheet, or a pleated sheet, or a helical shape. Then these secondary levels of protein structure are going to fold again. They fold in on themselves again and form the 3rd level of protein structure. And oftentimes it forms this kind of globular shape, but it might not. It might form a long linear shape, but it still is a 3rd level of structure. It's the 2nd time the protein is going to fold onto itself. And the 4th and last level of protein structure is called the quaternary structure, or the quaternary level, the 4th level. And this essentially is when you have one or more of these 3rd level proteins joined together with another 3rd level protein, right? Forming this large globular type structure in this case. So those are the 4 levels of protein structure, and you really need to make sure you know those, all right? And this kind of gives you a summary of how this all works. There is the primary structure, the secondary structures, and then these will fold again. You can see the helical shape here folding, and then a number of these join together to form the quaternary structure. Once a protein is formed, it can usually function if it's in its proper state, its native state, but anything that changes the shape of that protein completely changes its ability to be functional, and we call that denaturing. So if a protein is denatured, it cannot function. Some can renature, but most cannot. So if a protein gets denatured, it's done. And these are the different things that can cause denaturing of proteins. So pH, changes in temperature, and radiation, and certain types of chemicals. All right, so I'm going to stop here and we'll finish up talking about nucleic acids.